Yeah. Okay, so I'm going to talk a bit about our project that we've got ongoing, looking for biomarkers for disease and using proteins. So you already heard a bit about upper and lower motor neurons. So the upper motor neurons go from the brain down to the spinal cord and send signals from the brain, which then go to the lower motor neurons that go from the spinal cord out to the muscles and send signals for movement in the muscle. So MND involves both the upper and lower motor neurons, and the disease symptoms are quite variable across people, and it can have a bit to do with which motor neurons are being affected and in which region of the body. So the upper motor neurons are more responsible for symptoms of stiffness and a little bit of weakness, but the lower motor neurons are really the neurons that are responsible for the more disabling weakness and results in loss of muscle mass and muscle twitching. So here goes a picture of a healthy motor neuron. I stole this picture from a very nice series on YouTube called 2-Minute Neuroscience that explains how these things work. It's actually a very good video. I recommend looking at it. So you can see this healthy motor neuron. It's nice and spiky. This is a lower motor neuron going from the spine down to the muscle. You can see it's attached to the muscle, and the spikes at the end are quite embedded in the muscle. This is a less healthy looking motor neuron. The spikes are sort of limp there. The connection, the long string of it is less big. And you can see that the motor neuron's not really getting as much into the muscle, and the muscle's starting to waste a bit there. So what we're looking at is, this is my contribution to the picture, which also tells you why I stole the picture from YouTube. As motor neurons start dying with ALS, they can leak a bit. So the proteins that are in them can be released and they'll just go out into the bloodstream. So we want to look at whether we can detect these proteins and whether these are good indicators of disease onset and disease progression. So to do this, we're using samples collected as part of the SOLSA Systems Genomics Consortium. So this is a really big consortium across Australia that got launched along with after the Ice Bucket Challenge, and it is a way to bring together established ALS researchers, MND researchers, clinicians, and also brought in new researchers who have experience analysing large amounts of genomic data. And the main thing with this is there's multiple clinical sites across Australia, and everything's collected with a uniform protocol so that all our samples are comparable and available to researchers. So this is a blood sample, and what happens is it can get separated into components. This is one of the things they do to the blood samples that come in. So here we've got a blood sample that's been separated out into the red blood cells. We've got the white blood cells in the middle there. It's a very small layer. So that's all the immune cells. And the plasma. The plasma is normally a bit over half of the cells in the half the fluid in a blood sample. And so it contains no blood samples, but could contain these proteins from the motor neurons that are released as they are dying. So the way we're going to measure these is using a technology called Olink. This is a company, I think, come out of Sweden, and they provide panels that we can use to measure proteins in the blood. So we have actually used 2 panels. So we were funded to do the first panel, the neurology panel, which has 96 different proteins that are important for neurons in it. But actually, we managed to pull together some extra funding and also get another 96 proteins that are also quite important for neurology. We're going to do that in about 700 samples. So we have 300 people with ALS who have given a blood sample right when they registered into the SALSA study. So that's typically can be fairly early on in their disease. And then the same people again about 6 months later. It really depends when they came into the clinic and the spacing between, but average of about 6 months later. So we're getting 2 time points for these 300 people, and we're also getting 100 healthy individuals done. So this is currently being processed in Melbourne. The original plan was to do it in Sweden, but they got very busy, and a provider happened to open up in Melbourne while we were organising that. So that allowed us to do a lot more people and measure a lot more proteins. So that was great. What we intend to do with the data, so first we're going to look for differences between people with MND and healthy individuals, and look to see if we can pick up neuronal proteins in the blood at different amounts between people with disease and people without. And look at how these change with changes in disease progression. So these lines that are very hard to see are actually the 300 people with MND and their ALS-FRS scores as measured at 0. That's when they first come into the clinic, and the average of about 6 months later. There's quite a bit of variability in the time length there, but you can also see quite a range of different progression rates of disease between people there. So we're going to look at whether the proteins can predict the rate of prediction, and yeah, what the future disease progression is going to look like. Also going to join forces with a group in Sweden who have a similar cohort of people with ALS that are also going to measure this. So any results we get, we'll be able to confirm in another cohort. And we also have a Scottish cohort that have been measured for these proteins. And this is a healthy aging cohort of people about 70 or 80 years old. So we can see what it looks like in a healthy aging cohort and confirm that our signals are specific to ALS. So once we've done that, we really want to combine all our data sets that we've generated on the SALSA cohort. So this is called a systems genomics approach, where we're taking different levels all the way from your DNA, all the way to the small chemical modifications on the DNA, through to genes being expressed into proteins and into the metabolites, such as lipids levels, and bring these together to build a more complete story for the underlying biology in ALS. And while we've been waiting for the data to get generated for the protein levels, we've actually had a visitor from a group in Scotland who have used epigenetic data, so that's small modifications on the DNA, to predict protein levels. And we happen to have a lot of epigenetic data, so they've run it in our cohort, and it looks like we can see differences between people with ALS and not on the basis of that. So that gives me quite good hope that measuring the proteins directly will give us some good results. So what we're hoping to get out of this research is to be able to improve diagnosis. If we can measure protein in the blood that's very early on in disease, that would remove a lot of the long diagnosis period and would be a great improvement. Be able to predict progression, as you've seen multiple times now. The disease progression is different for everyone, and it would be handy to be able to have a marker that would allow us to better predict that. And understanding just the biology of the disease, because the more we understand the biology of the disease, the more potential targets for new therapeutic avenues are found. Okay, thank you.